You know that feeling, right? Whether it's aphids munching your prize-winning roses, or maybe those stubborn weeds just invading your vegetable patch.
Or even worse, a devastating disease sweeping through a whole crop. Pests are just a constant challenge.
Exactly. And for decades, our go-to solution was, well, often a spray bottle full of chemicals.
Hmm.
But we've all learned the hard lessons from that approach, the silent spring of DDT, pests developing resistance.
Right, and the ripple effect on everything from beneficial insects
Right.
to predatory birds and even, concerns for human health.
It's just clear we need a different way, a better way.
Absolutely. Those initial quick fixes from chemical biocides, they came with a whole host of long-term ecological consequences, often creating brand new problems.
Yeah.
And this realization has driven a really crucial shift in pest management. We're turning our attention towards what we call biological control.
Biological control.
Okay. And in this deep dive, we're going to explore a truly remarkable and I think often underestimated natural powerhouse, fungi.
Fungi. Okay, so imagine if we could leverage nature's own mechanisms to fight these battles for us against animal pests, invasive weeds, even other problematic diseases.
That's exactly the idea.
So our mission today is to explore how fungi are stepping into this role, how they're acting as these highly specific, environmentally conscious agents for biological control.
We'll unpack their surprising strengths, yeah, but also the hurdles to their widespread adoption.
And dive into some incredible real-world examples across these three major areas: animals, weeds, and other fungi.
And by the end, you should have a really solid grasp of this vital topic, all without needing a single visual aid.
Great. So let's start maybe with that history. The chemical pesticide story really is a bit of a cautionary tale, isn't it?
It really is. Chemicals like DDT, they initially seemed like miracle cures,
Right.
rapid broad-spectrum control for infestations.
Mhm.
But their persistence in the environment led to just devastating long-term problems. We saw predatory bird populations crash.
Right, that biological accumulation of DDT residues.
Exactly. And pests quickly developed resistance, making the chemicals less and less effective over time.
And even the newer generations of pesticides, maybe they're less persistent, but they're still often toxic to non-target organisms, right?
That's a huge issue. They can eliminate natural enemies, which can lead to outbreaks of secondary pests, or just a rapid resurgence of the original problem once you stop spraying.
So it's kind of a vicious cycle sometimes.
Precisely why the concept of biocontrol emerged as such a compelling alternative. Its core principle is elegantly simple, really.
Okay.
Instead of wiping out everything, you identify a natural predator, or maybe a parasite, or even a competitor of the target organism.
And then you encourage that agent to reduce the pest population naturally.
Exactly. Now, when we talk biocontrol, most people probably think of like ladybugs eating aphids or one insect fighting another.
Yeah, that's the classic image.
But this is where the story takes a fascinating turn. Fungi are, in many ways, potentially superior biocontrol agents compared to arthropods.
Superior. How so? What gives them the edge?
Well, consider the unique advantages. First, fungi boast an incredibly high reproductive capacity. They can produce just enormous numbers of spores ready to spread.
Okay, sheer numbers.
Second, they have a very short generation time. They multiply rapidly.
Quick turnaround. Makes sense.
Third, and this is really critical for precision, fungi are often highly specific in their action. They frequently attack only the host they've co-evolved with.
Ah, so less collateral damage, minimizing harm to non-target organisms.
Precisely.
Yep.
And finally, many fungi can form resting stages, or what we call saprobic phases. These allow them to survive for long periods when a host isn't available.
So they offer potential for more long-term control, not just a quick hit.
Exactly. It's a fundamental shift from that kill-everything-now chemical approach to a more restore ecological balance over time strategy.
Okay, given those incredible advantages, high reproduction, speed, specificity, persistence, I'm almost wondering, why haven't fungi completely dominated the pest control landscape already? What are the hurdles?
That's a crucial question. And it comes down to some significant challenges, especially under natural conditions. You see, a fungal parasite's population might build up, but often not quickly enough to control the target organism during the period when it causes the most damage.
Ah, okay. Timing is everything.
Right. So, for one, they might only damage rather than outright kill their host, which might not be enough sometimes.
Less dramatic than a chemical spray, maybe.
Two, they often reduce rather than eliminate the target population, which can be frustrating for people used to complete eradication.
Yeah, farmers might want zero pests, not just fewer pests.
And three, their effects can be relatively slow compared to chemicals. Humans, well, we're often impatient for results.
We like instant gratification.
We do. However, the fact that they're non-toxic, target-specific, self-reproducing, and self-perpetuating, these are powerful incentives to try and overcome those hurdles.
So how do we make it work then? What needs to happen?
To make fungal biocontrol work effectively, we need to manipulate several critical factors. Think of it like a checklist for success.
Okay, lay it out for us.
First, the fungus must be proven non-pathogenic to any economically valuable organisms. Can't have it killing the crop you're trying to protect.
Obviously, yeah, due diligence.
Second, a large amount of inoculum, that's the fungal material, usually the spores must be readily available. You need enough to start the process.
So production and supply.
Third, this inoculum needs to be properly distributed and early enough, ideally before the pest population really peaks.
Get ahead of the curve.
And finally, the climatic conditions have to be right. They must favor the fungus's growth, its sporulation, and its dispersal. Temperature, humidity, all crucial.
Right. So if any of those links in the chain are weak, the whole strategy might fail.
Precisely.
So it's clear fungi have this immense potential, but it's far more nuanced than just, sprinkling some spores around. It requires this delicate balance of biological understanding and environmental manipulation.
That's a perfect way to put it.
Okay, with that foundation, let's dive into some specific examples. Where are we seeing this in action? Maybe start with those organisms that compete directly with us for resources, animal pests.
Indeed, arthropods, especially insects, are arguably our greatest competitors. They damage crops, transmit diseases.
Yeah, the list is long.
Fortunately, a number of fungi are lethal parasites of these pests. We group them together as entomogenous fungi.
Entomogenous fungi. Got it. How do they actually work? Can you paint a picture for us since we don't have visuals?
I can try. Imagine a fungal spore landing on the host's cuticle that's its outer skin, its protective layer.
Okay, spore lands on the bug.
It's like a silent invasion. That spore germinates. It sends out a tiny, thread-like structure. We call it a germ tube.
Right, a little root.
Sort of. This tube acts like a microscopic drill. It penetrates right through the insect's tough, chitinous exoskeleton, that hard outer shell.
Whoa, it drills through the armor.
Exactly. Once inside, the fungus really takes hold. Its branching threads, known as hyphae, begin to proliferate, to spread. They start riddling the insect's internal organs, its viscera.
Oh man, so it's growing inside the insect?
Yes, consuming it from within. Eventually the host dies, and then often new spore-bearing structures sprout right out of its corpse.
Ew, but effective.
Very effective, ready to release fresh fungal material, that inoculum, to infect more unsuspecting pests.
That mechanism sounds incredibly potent, almost like something from a sci-fi movie, a microscopic alien invasion. So if that's the how, where have we actually seen this biological invasion successfully implemented in the real world?
Well, one of the earliest and maybe most historically significant examples is Boveria bassiana. You might see it sold under the trade name Boveria.
Beauveria bassiana. Okay.
In the early 1800s, this fungus caused something called muscardine disease. It absolutely devastated silkworm industries in Europe.
Ah, so it wasn't seen as beneficial then.
Not by the silkworm farmers, no. But an Italian scientist named Bassi discovered it was an infectious principle. He identified the fungus, that white powder on the mummified silkworms.
Right.
And his groundbreaking work back in 1834 actually laid the foundation for the germ theory of disease. A true milestone in scientific understanding, stemming from studying a fungus killing insects.
Wow, that's incredible. So how is it used today?
Today, Boverin is mass-produced. It's used in places like Russia to control the Colorado potato beetle, a pest notorious for just stripping potato plants bare.
Oh yeah, those are nasty.
It's applied twice, usually at about 1.0 to 1.5 kilograms per hectare. Each gram contains something like 30 billion canidia, or spores.
30 billion per gram. That's that high reproductive capacity you mentioned.
Exactly. And it's also effective against a whole range of other pests: codling moths, chinch bugs, grasshoppers, whiteflies, aphids, even termites.
Versatile stuff. What else is out there?
Then consider Metarhizium anisopliae, sometimes sold as Metaquino. This fungus has seen widespread use, especially in Brazil, against spittlebugs on sugarcane and pastures.
Spittlebugs, the ones that make that frothy stuff.
That's them. The nymphs create that protective, foamy home. But maybe its most compelling success story comes from the South Pacific. islands like Tonga Tapu and Western Samoa.
Okay, what happened there?
The devastating rhinoceros beetle arrived probably in the 1930s and just started wiping out coconut palms, a vital resource.
Oh,
But
no.
the introduction of Metarhizium, along with an entomopathogenic virus, successfully controlled the beetle by about 1968.
Wow, a tag team approach.
Yeah. And this allowed young trees to survive and the older trees to actually bear fruit again. It essentially saved a key economic resource for those islands. It also shows promise against things like mosquito larvae and even household cockroaches.
That's a fantastic turnaround story. Any others making waves?
Definitely. In Florida, there's Hirsutella thompsonii, known commercially as Mycar. It causes these spectacular natural epidemics among citrus rust mites.
So it occurs naturally, but we can boost it.
Exactly. Farmers now spray it early in the season to prevent those mite populations from building up and damaging the citrus fruit.
Smart. Proactive.
We also have Verticillium lecanii, which is available as a Vertalec and mycoinsecticides. This one causes natural outbreaks in plant-sucking pests like aphids and scale insects.
Aphids and scale insects.
Yeah.
Always tough ones.
They are. And what's interesting here is that different strains are tailored for specific targets. Vertalec is highly pathogenic to aphids, while mycoinsecticides is aimed at the notorious greenhouse whitefly.
Tailored strains. That really speaks to the specificity you mentioned earlier. It's not just one fungus fits all.
Not at all. The level of specialization can be quite remarkable.
What's particularly compelling here is just the sheer diversity of these fungi and the ingenuity involved in applying them. From mass spraying spores to understanding these really complex life cycles, it makes you realize how interconnected these natural systems are. It also seems like, some pests, particularly those scale insects and whiteflies, they seem especially vulnerable to these fungal attacks.
They often are, yes. And we're seeing promising newcomers entering the market too, like Paecilomyces fulmiceraceus. That's approved for use on ornamentals against whiteflies, aphids, thrips, spider mites.
A broad range for ornamentals.
And another one, Nomerea riley, shows high mortality in caterpillar pests, things that attack cabbage, clover, soybean. It's under intensive study right now.
Okay. Any others that are maybe more dramatic or harder to harness?
Well, there's Entomophthora. Many people might actually encounter this one without realizing it. It infects and kills houseflies.
Houseflies. Okay.
And the flies often exhibit this strange behavior. They crawl to exposed high locations before they die. And you might see this visible halo of discharged spores around the corpse.
Whoa, okay. A fungal halo. That's memorable.
It is. While it attacks various insects, its widespread use in biocontrol is challenging. The spores have a short lifespan, and it's very dependent on environmental factors, rainfall, temperature.
So less reliable, maybe?
It can be trickier to deploy predictably. However, it was successfully introduced in Australia to control the spotted alfalfa aphid, which is interesting because that aphid had arrived without its natural enemies.
Ah, the invasive pest problem. They leave their checks and balances behind.
Exactly.
Yeah.
That's a recurring theme in biocontrol, finding those natural enemies and reuniting them with the pest.
So it sounds like not all fungal biocontrol is about direct attack either, right? There are indirect methods.
That's a great point. Sometimes fungi work indirectly. For instance, there's a fungus called Phomopsis oblonga. It naturally occurs in elm bark,
Okay.
and it's been observed disrupting the breeding of bark beetles.
And bark beetles are vectors for?
Dutch elm disease, a really devastating tree disease.
Hmm.
So by interfering with the beetle's life cycle, the fungus helps control the spread of the disease.
Clever. Using the fungus to target the vector.
And then there's the truly tantalizing but as yet unfulfilled potential of Coelomomyces.
Coelomomyces, what does that target?
This is an obligate parasite of mosquito larvae, including Anopheles gambiae,
The malaria vector.
the notorious malaria vector. Despite causing heavy mortality in natural mosquito populations, its practical application is really complicated.
Why is that? If it kills mosquito larvae, that seems huge.
It does, but it requires an alternate host to complete its life cycle. A tiny crustacean, either a copepod or an ostracod.
Ah, so it needs two different hosts, like some rust fungi.
Exactly. You can't just spray the fungus. You need to manage two distinct populations in a specific sequence. It has a whole layer of complexity.
But the potential payoff.
Is immense. Given that mosquitoes cause, 7 million malaria cases and 500,000 deaths annually in Africa alone, mostly children, there are huge efforts underway to try and overcome this significant hurdle.
Wow. Okay, so fungi can be these precise assassins for animal pests, sometimes directly, sometimes indirectly. What about those persistent botanical invaders we call weeds? Let's turn our attention there. How are these microscopic allies revolutionizing the fight on that second major battleground?
Yeah, it's a completely different landscape, but the principle of biological control remains just as powerful, just as relevant. Weeds pose immense challenges, right? Competing with our crops for water, nutrients, light.
Plugging up harvesting equipment.
Exactly. Making life difficult for farmers, gardeners, everyone. Chemical herbicides, things like paraquat or 2,4-D, have been widely used.
A traditional approach?
But they've come with their own set of serious problems: toxicity, potential terrogenic effects, and often a general lack of selectivity.
Meaning they harm desirable plants too, not just the weeds.
Precisely. And that's where fungi offer a unique and really compelling advantage: their finely tuned selectivity. Many plant pathogenic fungi often restrict their attacks to just a single host species, or maybe a few closely related ones.
Much more targeted than a broad spectrum chemical.
Much more. So the classical biocontrol strategy for weeds involves searching the weed's native homeland.
Go back to the source.
Right. Find the specific fungal pathogens that keep it in check there, and then carefully introduce them. It's a much more precise and environmentally sound approach.
And we have some fantastic examples of this working in the real world, particularly with rust fungi. You mentioned they're very specific.
Incredibly specific fighters. Take Phragmidium violaceum. It's a European rust fungus.
Okay.
When European blackberries started encroaching on pastures down in Chile, becoming a real problem, this fungus was introduced. And it successfully suppressed the blackberry spread.
Wow. And just the blackberry.
Largely, yes. What's crucial here is that this rust is autoecious. That means it only needs one host to blackberry to complete its life cycle, so no other plants were threatened.
Autoecious. Okay, that's a key term meaning single host. That specificity is vital.
Absolutely. Then there's the really spectacular Australian success story with Petunia chondrolina.
Okay, what weed did that tackle?
This rust fungus, which came from Italy, was introduced back in 1971 to combat rush skeleton weed.
Rush skeleton weed. Doesn't sound pleasant.
It wasn't. It had been accidentally brought to Australia. It was just devastating hundreds of thousands of hectares of prime wheatland.
Oh, wow. A major economic impact.
Huge. But this introduction of Puccini chondrolina was so successful that nearly 500,000 hectares no longer need to be sprayed with chemical herbicides. It's estimated to have saved Australia an astonishing 112 times the cost of the program.
112 times the cost. That's an incredible return on investment.
It really is. And the ingenuity here truly shines when you consider a potential complication. On its home turf in Italy, Puccini chondrolina itself has a fungal hyperparasite.
A fungus that attacks the fungus?
Exactly, called Udar leucocarices. So the material introduced to Australia had to be meticulously checked to ensure it wasn't contaminated with this hyperparasite.
Because that would have weakened the biocontrol agent?
Significantly reduced its effectiveness. It shows the level of detail needed.
That example perfectly illustrates the careful research and the foresight required. You can't just release things without understanding the whole system.
Absolutely. Avoiding unintended consequences is paramount.
So classical biocontrol involves finding these natural enemies. But you also mentioned fungi being used like herbicides, right? Sprayed directly?
Yes, for many weeds, that's the approach. Fungal agents are mass-produced as mycoherbicides. They're sprayed directly onto the weeds, often in a massive inoculum.
Why massive?
The idea is to basically swamp any natural host resistance and initiate an epidemic, assuming the environmental conditions are right.
Ah, overwhelm the weeds' defenses. Any examples of these mycoherbicides?
Sure. Coletotrichum gluosporioides, sold as Colego, was actually the first patented practical mycoherbicide.
Colego, okay.
It controls northern joint vetch, which is a weed problem in rice and soybeans in the U.S. Sprays containing maybe 2 to 6 million canidia per milliliter can achieve 95% to 100% kill rates.
Very.
Another example is Phytophthora pulmivora. Now, this is technically an Oomycetes pathogen, sometimes called a pseudofungus, closely related but slightly different group.
Okay, Phytophthora.
It's the active ingredient in a product called Divine, used in Florida citrus groves to control strangler vine, which can, well, strangle the trees. It causes root and stem rot in the vine.
And how is that applied?
Just 1 pint of suspension, diluted in about 50 gallons of water, can treat an entire acre. And it persists in the soil from year to year, offering ongoing control.
Persistent control from a biological agent. That's impressive.
It is. And think about water hyacinth, that prolific aquatic weed that clogs waterways in tropical regions.
Oh yeah, huge problem in many places.
A fungus called Cercospora rodmanii was discovered causing a natural epidemic on it in Florida, after extensive research to ensure its safety for non-target plants and animals.
that crucial safety testing again.
Absolutely essential. It's now been patented and produced commercially as a mycoherbicide.
But it's not always straightforward, right? There must be cases where a potential fungal agent looks good but has issues.
Definitely. The story of another water hyacinth pathogen, Acremonium zonatum, highlights this perfectly. While it showed promise against water hyacinth,
What was the catch?
its host range wasn't quite specific enough. It was found to also infect valuable crop plants like figs and coffee.
Oof. Yeah, you can't release something like that.
Exactly. It underscores the rigorous testing needed for any biocontrol agent to avoid those potentially disastrous unintended side effects. It's all about that delicate balance.
But still, it's fascinating to see these other promising examples emerging too, like Colotrichum species controlling dodder in China and Phomopsis convolvulus being tested for field bindweed up in Canada.
The research is constantly expanding.
This really highlights that essential question. How do we ensure these highly specific agents don't cause problems down the line?
The meticulous research, the host
Yeah.
range testing, like what was done for Cercospora rhodmanii, it's absolutely key. It's how we avoid the mistakes of the past, those made with the broad-spectrum chemical herbicides.
Right.
And the idea that we can find these natural checks and balances, maybe ones that were lost when a weed invaded a new area, and reintroduce them, it just offers a far more sustainable path forward for weed management.
Okay, we've covered fungi versus animals, fungi versus weeds. Now, this next one might sound a bit like I don't know, a fungal fight club. Fungi controlling other fungi. Why would we want to encourage one fungus to attack another?
Ah, fungal fight club. I like that. It might sound counterintuitive, but there are actually three very logical reasons why this is a powerful strategy.
Okay, I'm listening. 3 reasons.
First, some fungi are naturally hyperparasitic. We touched on this briefly with the rust hyperparasite.
Mm.
They specialize in parasitizing other fungi, basically fighting fire with fungus.
Fungus eats fungus. Got it.
Second, fungi often compete intensely with one another. They fight for resources, for space, what scientists call the substrate. We can leverage this natural competition.
Survival of the fittest fungal addition.
Pretty much.
Yeah.
And third, we can use something called cross-protection or pre-inoculation. This is where introducing a non-pathogenic or maybe just a weakly pathogenic fungus can effectively immunize a plant against more virulent, more harmful strains of other fungi.
Like a fungal vaccine for the plant.
That's a great analogy, yes.
Okay, let's delve into some of these fungal battles then. Starting with the hyperparasites, the fungi fighting fungi directly.
Right. There's one called Sphaerolopsis phylum, the natural parasite of many different rust fungi, and is often credited with keeping their populations in check in nature.
So it's already out there working for us.
It is. And it's even being explored as a potential biocontrol agent against really serious blister rust diseases of pines, like Cronartium strobilinum, especially when the rust is on its alternate host, usually oak trees.
Ah, attacking the rust on its other host to protect the pines. Clever.
In one study, it infected something like 93% of the rust structures on oak leaves, significantly reducing the number of spores produced that could then go on to infect the pines.
Impressive reduction. Any others used commercially?
Yes, Shishinobola cisatia. This one is actually used as a spray to control powdery mildews, specifically Sphaerotheca, on greenhouse cucumbers.
Okay, targeting powdery mildew.
And in cocoa plantations, Cladobotrium amazonense helps control a serious disease called witch's broom, which is caused by another fungus.
Witch's broom sounds damaging.
It can be very damaging to cocoa production.
Okay, so that's hyperparasitism. What about competition? You mentioned fungi fighting for space and resources.
This is where the Trichoderma species really shine. They are true powerhouses in fungal antagonism. You might know them as common green molds, often seen in forest soils.
Trichoderma. Okay, the green molds. What makes them so good at competing?
They employ what you could call a double-barreled approach. They don't just compete for space. They actively parasitize the hyphae, the threads of other fungi. And they produce antibiotics that inhibit their competitors.
Wow, aggressive. Attack and chemical warfare.
Exactly. Trichoderma viride, for instance, is known to be effective against nasty soil pathogens like Rhizoctonia solani that causes damping off in seedlings and root rot, and also Armillaria mellia, a major root disease of trees.
So, protecting seedlings and established trees.
Then there's Trichoderma harzianum. This one is actually mass-produced and applied to soil to control Sclerotium rolfsi, another damaging fungus that attacks crops like tomatoes and peanuts.
Applied directly to the soil.
Yes, but there's a fascinating challenge here. Often, farmers still need to spray other fungicides for different diseases, like leaf spot on peanuts.
Right, multiple problems at once.
The issue is, those necessary chemical fungicide sprays can, unfortunately, reduce the trichoderma populations you've carefully applied.
Oh, so the chemical cure for one problem knocks out the biological cure for another.
Exactly. Which can lead to a resurgence of the sclerotium problem. It highlights the complexities of integrating different control methods.
Yeah, integrated pest management isn't always simple. What about using trichoderma for tree protection more directly?
Great application there. Applying trichoderma spores, often in maced or suspension, directly onto fresh wounds, like after pruning plum trees, can prevent infection by Asterium purpureum,
Which causes?
silverleaf disease, a damaging fungal disease of fruit trees. Similarly, panking freshly cut pine stumps with a trichoderma suspension prevents them from being colonized by Heterobasidian anosum.
And that's a serious root pathogen in conifers, right?
Extremely serious. In fact, there's a commercial alternative available specifically for this, using a different competitive fungus called Penniifera gigantea. But trichoderma works too.
So wound dressing with beneficial fungi.
Yes. Even things like spraying apple leaves with spores of Chaetomium globosum can reduce apple scab. And using it as a seed treatment protects corn seedlings against blight caused by Fusarium roseum. Competition works in many scenarios.
Okay, that covers hyperparasites and competition. What about that third strategy, pre-inoculation or cross-protection, the fungal vaccine idea?
Right, fungal immunization. We see some really innovative approaches here. For example, inoculating potting medium with spores of Tauromyces flavus.
Tauromyces flatus.
Doing that can reduce eggplant wilt, which is caused by Verticillium dahlia, by somewhere between 67% and 76%. And interestingly, it can also increase the yield of the eggplants by 18% to 54%.
Wow. Reduces disease and increases yield. That's a win-win.
Definitely. Another example, using weak, non-damaging strains of Verticillium albitrum. These have been shown to protect cotton plants from the more virulent, damaging strains that cause wilt.
How does that work? The weak strain triggers something.
Exactly. It stimulates the cotton plant to produce its own natural antifungal compounds, chemicals we call phytolexins, so the plant boosts its own defenses.
The fungus triggers the plant's immune system, basically.
That's a good way to think about it. Similarly, Verticillium nigrescens, which doesn't cause much harm, can induce resistance in mint plants against the serious wilt-producing Verticillium dahliae.
Fascinating. It's like training the plant to defend itself.
And consider peach canker. This is a devastating disease caused by a fungus, Cytospora, which often gets in through pruning wounds or other injuries.
Another wound invader.
Yes. And here, using competitive fungi like trichoderma and another one called gliocladium, shows great promise as a preventative treatment for those wounds.
Prophylactic protection. But you saved a really interesting one for last, didn't you? Something about strawberries and bees.
Ah, yes. This is a truly ingenious application for controlling strawberry gray mold, which is caused by the fungus Botrytis cinerea, a major headache for strawberry growers.
Green mold. Yeah, it can ruin berries quickly.
So researchers developed a system using a mycoparasite, a fungus that attacks botrytis, called Gliocladium roseum.
Okay, Gliocladium roseum. How do they apply it? Not spraying, presumably.
No, this is the brilliant part. They designed a special dispenser for the beehive entrance. As honeybees leave the hive to forage,
Yeah.
they walk through this dispenser and are automatically dusted with about 50,000 Gliocladium canidia each.
The bees get dusted with the good fungus.
Exactly. And where do bees go?
Straight to the flowers.
Precisely. They deliver these beneficial fungal spores directly to the strawberry flowers, which is the crucial stage where the botrytis infection typically begins.
That is absolutely brilliant, using the bees as targeted delivery drones for the biocontrol agent.
Isn't it? It's a perfect example of thinking outside the box, or in this case, outside the chemical spray bottle.
Huh.
It really drives home the point about the incredible adaptability and specificity we can harness with these fungal agents.
It really does. Wow.
And if we connect this back to the bigger picture, all these examples, from insects to weeds to other fungi, they really show us that there's always more to learn from nature. And that integrated pest management programs carefully combining these fungal agents with other strategies, maybe cultural practices or resistant varieties, are likely the most effective and sustainable way forward.
What an incredible journey through this unseen world of fungal allies. It's really opened my eyes. We've seen how fungi are revolutionizing biological control in these three huge areas: directly fighting animal pests, controlling weeds that threaten our crops, and even battling other harmful fungi in these complex interactions.
Precisely. And as our understanding grows, our understanding of the adverse effects of chemical biocides, both on the biosphere and, frankly, on ourselves, the search for natural, sustainable alternatives like fungal biocontrol becomes increasingly urgent and exciting.
Yeah, exciting is the right word.
These microscopic powerhouses offer a path towards a more balanced, more resilient ecosystem, moving us away from that kind of broad spectrum chemical warfare towards a more precise natural defense system.
So maybe the next time you're dealing with a pest problem or just thinking about agriculture or gardening, consider this. What other tiny, unseen allies might be waiting right there in nature, ready to help us cultivate a healthier, more balanced world?
It's a compelling thought.
Thank you so much for joining us on this deep dive. We really hope you've enjoyed uncovering the hidden and, frankly, amazing power of fungi with us today.